Okay, so here we are with chapter 21. This is the beginning of our final unit. So from these chapters here on out, this is what your final exam will be on. It will not be cumulative. It'll just be the chapters in this unit. And I think I posted the study guide for this unit already. This picture that you see on the first slide is a picture, it's called a rouleau. I think that's like a gambling term, isn't it? Like a game? I don't know. Anyway, so this is a rouleau. It's a single file line of red blood cells going through a capillary. So we're going to start this unit talking about blood, because then we'll get into cardiovascular system and heart. We'll look at the composition and functions of blood, and then we'll really spend a lot of time talking about blood types. So we each have our own specific blood type. So these are the components of blood. And you should know how to label this diagram in your notes, and I'll eventually show you this picture. So in your notes, there's this diagram, and I'll show you that picture. You should know how to label that. That'll probably be a test question. The erythrocytes make up about 44% of a blood sample. That's the lowest layer on that vial of blood. The buffy coat is the middle layer containing all leukocytes, which are your white blood cells, and platelets, which are involved with helping your blood to clot. That forms about 101% of your blood sample. And then the plasma is the straw-colored liquid that makes up the top part of that blood sample, about 55%. The formed elements of blood, so these, we call them the formed elements of blood, are the erythrocytes, leukocytes, and platelets. So those are red blood cells, white blood cells, and platelets are called the formed elements, because those are the ones that are formed in your body and are made by your body itself. Okay, so this is a look if you go to a lab and they take a sample of your blood, what do they do with it? And what are they testing for? So they take a vial of blood out of your arm, and then they put it in a centrifuge, which spins it extremely fast. Some of you guys might have done this if you work in a clinical setting. And then when it's spun really fast, it separates the blood sample into these parts that we just talked about. So the first part is your plasma. Most of plasma is made up of water. I don't know if I've told you my stories of donating plasma in college for money, but probably not the best way to make money. But a lot of people do it because
I've done it.
You've done it. Okay, yeah. Is it popular now still? I don't know.
Yes.
Okay. Yeah, you could make $200 or $300 a month.
Yeah. Well, especially right now, they need plasma.
Oh, good. See, so you're helping people, and you can make some money. I don't know. I don't know if I recommend it, because I still think it messes with. I don't know. We'll keep moving on. You can do as you want with your body, but anytime you sell your body parts, I just don't know about that. Anyway, plasma is mostly made up of water, proteins are in plasma, and other solutes are in plasma. Then the buffy coat is the middle layer that separates out here the plasma from the red blood cells right below it. And that buffy coat is where all your platelets are found and where all your leukocytes are found. Your leukocytes, you have 5 different types of leukocytes. Those are your white blood cells. I should probably teach you more about those, but I don't. But each type of leukocyte has a particular function, and your white blood cells help with the immune response. So they help to fight off disease pathogens in the body. So these little tiny cells will engulf anything foreign and try to get rid of it. Then the erythrocytes make up the bottom 44% of your whole blood. So this is why when someone tests your white blood cell or your leukocyte count, they're getting this vial of blood sample and then taking a syringe to take off that buffy coat layer and then to test your white blood cell count. So that's a whole blood sample, the separation of it, and the different layers. So you should know those different layers. Then the 3 functions of blood are your next part in your notes. Blood helps with transportation, regulation, and protection. So the transportation of oxygen and carbon dioxide. All your cells, all the processes in the body need oxygen. And carbon dioxide is always produced as a waste product in the process of creating ATP, which you'll learn about in physiology. It transports hormones. So hormones get transported through the bloodstream. Also nutrients, wastes, and then regulation. Blood helps maintain temperature, pH levels of the body, which is extremely important, fluid levels in your cardiovascular system, protection. Blood has those leukocytes in that buffy coat layer, so the leukocytes will guard against any sort of infection by triggering an immune response to antigens. And an antigen is any substance perceived as foreign, if it's different from the antigens that you already have yourself. Blood carries antibodies, and antibodies will attack specific antigens that it perceives as foreign. So the formed elements consist of erythrocytes, leukocytes, and platelets. And again, formed elements are called formed because they're what your body is making, formed elements, not any proteins that you're taking in, which would be in the plasma. So basically everything except the plasma is the formed element. The formed elements can be viewed with a blood smear, and this is something we could do in lab. I've done it in other labs, but wouldn't have done this here. You can actually withdraw or prick your own blood, place a sample of it on a slide, use a stain, and then you can look at and see the red blood cells and then the different leukocytes. And you can see the different leukocytes here. You probably recognize a picture like this from when we studied it in histology. Okay. So then we'll talk a little bit about the hematocrit. The hematocrit is the percentage of the volume of all formed elements in the blood. And because formed elements are mostly made up of erythrocytes, red blood cells, clinically it's just the percentage volume of erythrocytes. The hematocrit can vary with age and your sex or gender. Children can be a little more variable, and males have a slightly higher on average hematocrit or percentage of red blood cells than females. You can see here the general size, the function, the lifespan, and then the density in blood of each of the formed elements. Okay. So we'll start with erythrocytes. These are commonly, the prefix erythro means red, and cyte means cell. So these are commonly called red blood cells. They're not true cells because they lack a nucleus and all organelles. So they're specifically devised or created, you're made to look like this biconcave disc shape. So kind of like a donut without the hole in the middle. And they're created that way so that they can withhold as much oxygen as possible. If it gets too loud, I'll move into my garage. Sorry, guys. As they pass through small blood vessels, they line up in a single file line termed a rouleau. So this is what a red blood cell erythrocyte looks like. It has this biconcave structure, so like a donut, but with the middle still intact. And no nucleus, no organelles. The only thing they carry are hemoglobin molecules, and each red blood cell carries about 250 to 280 million hemoglobin molecules. And a hemoglobin is a protein that carries oxygen. And there's a picture from the first slide. So here's where we'll talk about hemoglobin. Every red blood cell contains about 280 million molecules of hemoglobin. Hemoglobin can be oxygenated or deoxygenated. It's oxygenated if it's maximally loaded with oxygen, and it's deoxygenated when some oxygen is lost and carbon dioxide is gained, usually when your blood is carrying carbon dioxide as a waste product to get rid of it. Each hemoglobin molecule consists of 4 what we call globin polypeptides, alpha and beta chains. Those 4 globin polypeptides, you don't have to know what they look like, but because each hemoglobin has 4 chains, each chain can hold 1 oxygen molecule. So every hemoglobin molecule can hold 4 oxygens and carry that oxygen. So each of the 4 globin chains possess a heme group, which contain an iron right in the center of it for oxygen binding. So thus every hemoglobin molecule can bind a total of 4 oxygen molecules. So if we think about in the body, if your body has, or if each red blood cell, excuse me, has 280 million molecules of hemoglobin, and each hemoglobin can carry 4 oxygens, one red blood cell can carry 1 trillion molecules of oxygen, which is just so much. It's crazy to think about it, but it shows you how important oxygen is in the body. Let me know if you have any questions as you're going, or type them in the chat. The erythrocyte life cycle, I think this is a test question to these ones that are with the asterisk. Erythrocytes have short lifespans, so they stay in your bloodstream about 120 days. They're phagocytized, which means they're broken down and kind of engulfed and, you know, rejected as waste in your liver and spleen. The heme group is converted to biliverdin, which is then converted to bilirubin, which just becomes part of bile for your digestive system. The iron then will be transported to the liver and stored until more erythrocytes are created. And erythrocyte proteins are broken down into free amino acids. So this is just a look at how red blood cells are recycled. They're created in the red bone marrow of your bones, if you remember that. Then they maintain their circulation for about 120 days, and then they'll get broken up in the liver and spleen into their kind of key molecular components, and then they'll be recycled for future use. Okay, then we get to blood types, which is everyone's favorite. This is kind of one of the trickier parts about. So blood types, how many of you know your blood types? Some of you do? Okay. So you're good, okay. If you don't, that's okay. You'll probably learn it when you have a major surgery or something. Or if you donate blood, they'll let you know your blood type there too. So how do we get particular blood types? Well, every single red blood cell that is in your body has what we call a surface antigen. And this antigen is some sort of protein or sugar that's attached to the red blood cell. And that antigen gives your erythrocyte the blood type name. These surface antigens are in the ABO blood group, and the presence or absence of A and B surface antigens is what determine your blood type. So for example, if you're type B blood, your erythrocytes have only the antigen B on the surface. If you're type O blood, you don't have either A or B antigen. So basically you can be A, you can be B, you can be AB, or you can be type O. And the AB and AB just refer to what surface antigens, and again, those are little proteins that are attached to your red blood cell. And if you're type O, you do not have either antigen attached. So I am A positive, and we'll talk about where the positive and negative come in. But first, so these specific surface antigens present, they're also accompanied in the plasma by antibodies. And these are proteins in your immune system that will attack antigens that are foreign to that individual. So type B blood has antibodies against A, so we call them anti-A antibodies. Type O blood has both anti-A and anti-B antibodies in its plasma. Type AB blood, because type AB blood has both surface antigen on its surface, A and B, type AB blood doesn't have any antibodies because it wouldn't create an antibody that would protect an antigen that it has on its surface. Okay? How do we feel about that so far? Hopefully okay. We'll keep going here. If mismatched blood is transfused, the recipient's antibodies will hook on to the antigen that's foreign, and it'll cause agglutination, which just means it'll cause clumping of that blood. And the clumping of those erythrocytes can lead to hemolysis, or the red blood cells splitting. So let's take a look at this picture here. So if we just look at type A blood has the A surface antigen on its red blood cell surface, and it will create anti-B antibodies, which will hook up to surface antigen B. So you can see there's little purple triangles indicating that the surface antigen B will hook onto those anti-B antibodies. So it will kind of defend the body against any Bs. Type B blood has the surface antigen B on its surface, and it creates anti-A antibodies. Type AB blood has both surface antigens, A and B, but it doesn't create any antibodies because it won't create antibodies that attack itself. And then type O blood has neither surface antigen A or B, so it will create both types of antibodies. Then where do we get this positive and negative? The positive and negative comes from another antigen called the rhesus factor or antigen D. So everyone has an A, B, O blood type, and then they have an either positive or negative. And the positive and negative just refers to the presence or absence of the Rh antigen. So if you're Rh positive, I'm A positive, so I'm Rh positive. I have this extra surface antigen D on my red blood cells. I do not create any anti-D antibodies unless I'm exposed. Well, I wouldn't create anti-D antibodies because I have the surface antigen D on my red blood cells. If you're Rh negative, you don't have the surface antigen D, so there's just an absence of that antigen. And you don't create anti-D antibodies unless you're exposed to Rh positive blood, which can cause problems in pregnancy. So if you're pregnant, your doctor will ask you what's your blood type. Well, they'll figure that out, and we'll talk about that in a second. Okay, so just in really review quickly, the type of blood you are just refers to the presence or absence of the antigens on the surface of your red blood cells. So if you're type A, you have A antigens, B, you have B, AB, you have both, and O, you have neither. Positive and negative refers to the absence or presence of surface antigen D. So if you're B positive, you have type B antigens and D antigens. If you're O negative, you don't have any antigens, and you're negative, so you don't even have the D antigen present. Okay? That's kind of quick. All right. This is what happens in a bad blood transfusion. We call this agglutination. So if we have a donor and a recipient blood type, let's say we have a type A blood of donor matches with a type A blood of a recipient, which contains anti-B antibodies. You can see that the antibodies do not cause any clumping because they don't fit in. They don't match up with the type B antigens because there's none present. So A and A mix together, and it's fine because the antibodies don't kind of clump on to the red blood cells. So in lab, we can see this and test for blood types that are compatible, and you wouldn't see any clumping. You would see no agglutination. So that's a good blood transfusion. If we have a bad blood transfusion and we have a donor type A blood plus a recipient of type B blood, you can see how the type B blood recipient has anti-A antibodies in its plasma that are directly kind of perfectly aligned to hook on to those type A antigens of the type A person. So when it does that, when it hooks on those antibodies to the antigens on the red blood cell, that causes the red blood cells to clump, and we call that agglutination, and that can be cause for a bad blood transfusion. And then clumping can lead to hemolysis. So hemolysis is when your red blood cells split. If your red blood cells split open, they lose their oxygen, your cells can't get oxygen, and you'll die pretty quickly. So bad blood transfusions are not good. This explains the Rh factor. It's determined by the presence or absence of that surface antigen D. So when it's present, you are positive for it. And anti-D antibodies can only occur when an Rh negative individual is exposed to Rh positive blood. And a person who has all three antigens on their red blood cells would be A, B positive, because they'll have A, they'll have B, and then they'll have that D surface antigen. So Rh negative, when is this ever a problem? So in pregnancy, if I was A negative, or if a mother is Rh negative, um, she doesn't have that D antigen on her red blood cells, and she doesn't have antibodies against it until she's exposed to Rh positive blood. So she could have a baby with Rh positive blood thanks to her partner or the baby's dad. That when she's exposed to that Rh positive baby, then she'll create antibodies against her baby's blood type. And it doesn't really cause any complications with the first pregnancy because their bloods don't really mix until delivery, but it can cause complications for future pregnancies that this Rh negative mother could have, because now she has antibodies in her system against the Rh factor. So if she has other kids who are Rh positive, then that could be a problem, because if there were to be a mixing of blood, those antibodies would attack the fetus's red blood cells. Okay? If we were in class, we'd look and we'd have you guys go up to the board, and we'd look at who has A positive, A negative blood. And then we're going to talk about who people can receive and donate from. And I have a really easy trick to remember this. So I'm going to go right away to, we have one chat. You're A positive too, Erin. All right, we're A positive sisters then. Okay, so here's a look at, this is just, you can find these on the internet. That's probably where I found this. So who can you give blood to and who can you receive blood from? I learned this from another professor, I think last semester at another school, and I just thought it was really easy to tell the difference. So if you guys can remember this, you can donate to anyone with the same antibodies or more, and a particular blood type can receive from anyone with the same antibodies or fewer. So a particular blood type can donate to anyone with the same antibodies or more, and they can receive from anyone with the same antibodies or fewer. Those should be antigens. I made a mistake there. Let's do that before I get more confused. Okay, so let's look at type A positive. So if you're A positive, you can give blood to, you can donate, so donate more to anyone with the same antigens that you have. So you can give to A positive, or what's the only other antigen you can add to A positive? B. So you could donate to an AB positive person. You couldn't donate to a B positive or B negative person because they don't have the same antigens. A positive people can receive blood from anyone with the same antigens or fewer. So A positive, A negative, O positive, or O negative. And you can work your way down the line. And I don't know, that really helped me clarify it with some students in the past. So maybe that'll help you. You can donate to anyone with the same antigens or more, and a person can receive from anyone with the same antigens or fewer. Some blood types are extra special. And I usually like to pick on these people, not pick them out, but just make them feel special in class, because if you're AB positive, you're considered the universal recipient. So if you're AB positive, you can receive from anyone with the same antigens or fewer. So remember, AB positive blood has all three antigens on the surface. And if you're AB positive, you're not going to create antibodies against yourself. So because you have no antibodies in your blood, you can receive blood from anybody because you don't have any antibodies that would attack anyone else's blood that you'd be receiving it from. So AB positive people, we call them universal recipients. Universal recipient is AB positive. So if you're AB positive, you're gonna outlive us all, like zombie apocalypse or something. The other special one is O negative. If you're O negative, you're considered the universal donor, so you can give blood to anyone. So again, If you're O negative, you can donate to anyone with the same antigens or more. Remember, O negative, you don't have A antigens, you don't have B antigens, and you don't have the RH factor. So because of that, you can be given to anyone because anyone won't the antibodies won't be able to clump up to any antigens that you have on your surface. So O negative is considered the universal donor. Okay, moving on. How are you guys feeling about that? I know that's a long explanation, but that's kind of the bulk of why this is a shorter lecture. We'll briefly kind of finish the lecture talking about some ways that leukocytes, platelets form. Leukocytes are your white blood cells. They possess a nucleus and organelles. They're larger than your red blood cells. These are the ones, remember, that help initiate an immune response to defend the body against pathogens. There's some pathological conditions that can cause abnormal numbers of leukocytes. So leukopenia is a reduced number of cells, and leukocytosis is an elevated number of cells. There's 5, I'm just gonna skip down to the 5 types of leukocytes because this is what you might see again in future classes. The 5 types of leukocytes, they either have granules in them or they have a lack of granules. And the granulocytes include neutrophils, eosinophils, and basophils. And agranulocytes include lymphocytes and monocytes. So those are the 5 types of leukocytes, white blood cells, and they each have their own particular function. So these are the 5 types. You can see that agranulocytes don't have these little tiny kind of grainy materials in their cell, so they look more clear. And the granulocytes look like they have little tiny specks of granules in them. So these are your 5 types of leukocytes. Here's the 5 types and their functions. Again, I don't really test you on this because there's a lot in this unit. Maybe I should. Future anatomy class. Future anatomy, not for you guys. Okay. Then platelets are small, irregular, membrane-enclosed cell fragments. They do not have a nucleus. They're produced in your red bone marrow from cells that we call megakaryocytes. And these are the cells that are involved in blood clotting. So if you get cut, your platelets will go to that site where you got cut, and they'll try to kind of hole up and stop you from bleeding. Thrombocytopenia is an abnormally small number of platelets. Blood thinners like heparin, warfarin, my dad's been on Coumadin for years, they slow down your body's process to make clots. So they slow down the process of these platelets to go to the site of damage, just so that your blood can get through. Usually people with high cholesterol, heart disease are on blood thinners. So it's prescribed to those susceptible to heart attack, stroke, or cardiovascular disease, people whose arteries are kind of closed up, to try to keep the blood thin and keep it moving. Platelet formation, you can see here, they're created from pieces of megakaryocytes, and the megakaryocytes will then be in the blood, and they'll go to a site of damage to try to keep the blood or help the blood to clot. This is what a blood clot looks like. So the platelets produce these fibrin materials that kind of clot the blood. And again, clotting the blood is important in areas of damage. If you need to stop bleeding, it'll form this clot. What's scary then is if this clot forms and then it travels to parts of the brain or your lungs, and then it sort of blocks your arteries or areas in your lungs. Well, you need to know that platelets are called thrombocytes. Yes, that's a thrombosis. Yeah, you should be familiar with, because you guys are all entering medical field. I don't know if I asked you about that, but you should know that platelets are called thrombocytes. Yes. Okay. Formed elements are continually produced in your red bone marrow, and we call the forming of red blood cells hemopoiesis. It begins with a pluripotent stem cell called a hematocytoblast, and these hematocytoblasts are like they're stem cells, so they can go one way or the other. The myeloid line will form one set, and the lymphoid line will form your lymphocytes. I won't ask you again too many questions about this. Hemopoiesis involves several colony-stimulating factors, and they just increase the formation of erythrocytes and other formed elements. You guys don't need to know all these colony-stimulating factors, but they just stimulate the production of a particular cell type. So this is a look at that hemocytoblast, which is the blood stem cell, and how it will differentiate into a myeloid or lymphoid line, and then go on to form the different types of leukocytes, lymphocytes, platelets, or erythrocytes. So all of your different formed elements, whether they're a platelet, an erythrocyte, or a leukocyte, will all come from the first stem cell. Erythropoiesis produces red blood cells. It happens at about 3 million per second. The rate is controlled by a hormone called erythropoietin. Again, I'm going to kind of skip over the rest of this, but erythropoiesis is the production of red blood cells. Thrombopoiesis is the production of platelets, also thrombocytes. And again, they're formed by that megakaryoblast that comes from that first stem cell line. And each megakaryocyte can produce thousands of platelets as they break off from it. Leukopoiesis is the production of leukocytes. They will mature into granulocytes, monocytes, or lymphocytes, and those are just different types of leukocytes themselves. And that's all I have for this lecture, guys. So I'm just going to stop the recording here.